Okay, today we're going over the lungs, membranes, pressures, mechanisms, how negative pressures work, what a pneumothorax is, surfactant, etc. And then finally, we'll kind of discuss a little bit how oxygen and carbon dioxide travel within the blood and the different mechanisms for that, as well as dissociation curves for hemoglobin. So let's jump right into it. This is your lung. You have a couple membranes here. You have a parietal and a visceral membrane of the lung. And within that cavity, you have a pleural cavity that's filled with serous fluid. And what that's going to do is create a negative pressure. This fluid is going to be like it's like kind of like a suction cup. It's going to pull the lung to the cavity wall, right? So a couple of those. So that's important to know when we get into negative pressures and ventilation in inspiration, respiration, expiration, all that. So there's a couple layers of the membrane. This is within the lung itself. So if we had, well, let's look at here. We have an alveolus, right? Just one of them. And there's going to be 6 layers that branch out from here. So 6 layers. And that's going to be the fluid covering the alveoli, which is surfactant. And so it's going to kind of cover this inside. Then you're going to have an epithelial cell, squamous cells, a basement membrane, alveolar epithelium. Then you'll have a little bit of space between your capillary basement and, well, yeah, you'll have that space. Then you'll have your capillary endothelial basement, and then you'll have other squamous cell walls of capillaries. So if we look here, there's 6 layers. They just kind of go out. If we're going from internal to external, these are all within the alveoli. And those membranes are made up so that we can actually breathe. If you see, we have capillaries surrounding your alveoli, and that allows for gas exchange between your blood and the capillaries and the gas in your alveoli. There's a couple of cells within the alveoli. Type 1 pneumocytes, they do most of the gas exchange, and type 2 do surfactant secretion. What is surfactant, you might ask? Well, let's turn to here. Surfactant is just a hydrophobic and hydrophilic lipoprotein. So what it's going to do is within your alveoli here, you'll have water, and water has surface tension. So if you've ever seen a water skipper, they kind of float on the top of the water. They're just acting on that surface tension or cohesion of water molecules. And so surfactant increases tension within the alveoli to collapse. And we don't want our lungs to collapse, so in order to minimize that effect, we come up with something called surfactant, which spreads out that cohesive effect of water and regulates how much we want to deflate our lungs and how effective we are in pulling the walls in together. So if we had a lack of surfactant within this diagram, we can see, well, first off, let's cover Laplace's law. Yeah, Laplace's law. So this is kind of a mathematical scenario. This is pressure to pull in. This is, this is pressure that the alveoli sustains and how hard it is to get in air. And this is proportional to our surface tension. So while we have elasticity of the cells of the alveoli and they're pulling in and wanting to collapse, we also have the surface tension of the water, and they're proportional. So if you increase surface tension, then you have a greater pressure to collapse that alveoli. And this is dependent on how large your alveoli is. So if you increase the radius, then the water molecules are spread farther apart so they can attract each other, so you're less likely to collapse. You have a large R, low pressure to collapse. But say you had a small radius, then water's going to attract and it's going to want to collapse this alveoli. You'll have high pressure to collapse. With surfactant, though, you reduce the surface tension and you equalize this airfill between your bigger alveolus and your smaller alveolus. Equal pressure, even though you have different radii within each alveoli. I don't think I explained pneumothorax yet, but yeah, that's surfactant for you. A pneumothorax, as I was explaining earlier, you had your lung, right? And you had that serous fluid in the middle, and you had your cavity wall, right? So these two are kind of sticking together by suction. There's a vacuum effect. Say you had a pneumothorax. What happens is air gets into this cavity and the negative vacuum pressure isn't pulling so much on the lung, so your lung is going to deflate because it doesn't have a vacuum effect to pull it out again. Another way this can happen is if a lot of liquid enters into this cavity too. If you separate the lungs and the cavity wall too much, then there's no suction or negative pressure allowing you to pull the lung out. So that's your pneumothorax. Let's back up a little bit. There was something else I wanted to discuss. It was, this. This is pressures. So you have a couple things that go into that. If you want to exhale, you have a diaphragm. So let's go back here. We have a diaphragm. If you want to inhale, this diaphragm goes down. If you want to exhale, that diaphragm relaxes. And so that's one mechanical way that our muscles can regulate whether we inspire or expire. Some things we need to keep in mind is atmospheric pressure is usually set at 0. And so in order to inspire, we need to create a negative pressure within the alveoli. So you would pull in air. Pressure always goes from high to low. So if we have a 0 here at atmospheric pressure and a negative here in our alveoli, then we're going to be pulling in air. To expire, we change that and we increase the pressure within the alveoli, and that causes us to expire because we've exceeded atmospheric pressure. We are now high in the alveoli and low in the atmosphere, so we breathe out. Pleural pressure, that's kind of that vacuum effect I discussed within this pleural cavity. That is created by lymph vessels. So lymphatic vessels are pulling fluid out of this, or serous fluid out of the pleural cavity, and that creates a -4 millimeters of mercury pressure. So if we want positive pressure ventilation, we're going to force air in. We're going to cause a high pressure outside, low pressure inside. Negative pressure ventilation, we actually make the pleural cavity more negative, and that's going to pull our lungs out, decreasing the pressure inside the alveoli, and thus air is going to flow in. So if we're talking about transpulmonary pressure, that's just the difference between pleural pressure, that's our negative, and alveolar pressure up here. Between those two, we can have transpulmonary pressure. Okay, finally, we're gonna describe blood transport within the body. So none of this works if we don't have carbon dioxide or oxygen to work with. Within blood, the majority is going to be transferred or converted into bicarbonate so that it's not in its gaseous phase. So carbon dioxide, small, it won't bind to hemoglobin a lot. It doesn't travel in the blood. It's mostly converted to bicarbonate, and that allows it to travel in the blood freely. Oxygen, on the other hand, is taken up by hemoglobin. The majority of it is. So if we're in the lungs, and you have a bloodstream here, right? We're gonna have a bunch of bicarbonate flowing through the blood, transferring our CO2, and then once it hits the lungs, it's gonna transfer into there and convert back to CO2. O2, on the other hand, is gonna have a high pressure outside, and it's gonna want to flow into our arteries or arterioles, because we have a high pressure in here and a low pressure in here. And what's going to transport that? Hemoglobin has 4 subunits, and that will bind to oxygen and transfer it throughout the blood. Some of the things that can well, actually, let's go over here. You'll have partial pressures of gradients in the lungs. I kind of explained that over here with this picture. If you have a partial pressure of O2 in your alveoli of 104 and in the capillaries of 40, so if we're in here, we have veins, we've got arteries, 104 to 40, high to low, we're going to want to move oxygen into the arteries. Well, really, it's going to bind to hemoglobin and be transferred throughout the blood. If we look here, as it deoxygenates and hits tissues, we're going to drop to 95. We're gonna, in the interstitial spaces, we're gonna have a oxygen pressure of 40, so it's gonna continue to drop, and then our cells are gonna have a 20 partial pressure of oxygen, and so that's going to allow oxygen to diffuse all the way from 104 high outside pressure all the way into our cells, which are low at 20. Carbon dioxide is not as drastic, and we're trying to get it out of the body. So within the cells, we'll have a concentration or a pressure of 46. Interstitial space will have 45. In the blood, we'll have 40. That 40 will travel, within the blood, we'll have 45. And then once we hit the lungs, we'll have a 40 partial pressure in the lungs, and it will want to dissociate from hydrogens, and bicarbonate will convert into carbon dioxide. So that's transport of CO2 and oxygen in the blood. And let's go over to our final part of this essay question. This is the oxygen hemoglobin dissociation curve. A couple factors that influence the affinity of hemoglobin for oxygen is going to be pH, carbon dioxide concentrations, temperature, as well as BPG. A good way to remember that is cadet or cabet for BPG. Let's say this is the Bohr effect. pH, well, as I mentioned earlier, you have hemoglobin and it'll bind oxygen. You have T states and you have R states. In the T state, you're gonna have a low affinity for oxygen. Oxygen is gonna dissociate from hemoglobin at that point, and an R state is gonna accept oxygen. So hydrogen, if it binds to hemoglobin, it induces that T state, which pushes off oxygen. So if we scroll over here, if you have a low pH, you have a lot of hydrogens binding to hemoglobin, and that's going to induce a T state and push off oxygen. In contrast, if you have a high pH, which means not a lot of hydrogens are binding to hemoglobin, then you'll have a higher affinity for oxygen, or more R state hemoglobins for oxygen to bind to. Another thing, if you have a high pressure of oxygen, of course, you're going to force oxygen into the blood and increase binding of it. In contrast, if you have a low O2 in the blood, you'll actually release more. You'll enter that T state of hemoglobin, and you'll release more oxygen because you need it. It senses, oh, we have low oxygen, we need to release more and oxygenate the hypoxic cells. So these factors can constitute a right shift. So if we have our hemoglobin oxygen dissociation curve, then a right shift would be a low pH. So we had a right shift, low pH, less affinity for oxygen. So of course our percentage of oxygen is going to go down, and our partial pressure of oxygen goes down as well within the blood. Another thing, if carbon dioxide increases, then we have something called the Haldane effect, where if you bind, or no, that's opposite. If you increase CO2 or pressures, then it'll bind more readily to hemoglobin, and that will kind of bump off oxygen, creating less affinity for oxygen to bind. Increasing of temperature, more activity, more heat, increasing of viscosity of fluids will bump off oxygen as well. So colder air will hold a little bit more oxygen, or at least your body will hold more oxygen than colder air. And then BPG is kind of interesting. It is used primarily in placenta, and if a baby's placenta has a higher BPG, we've now lowered the affinity of the mother's blood for oxygen, and so that oxygen will transfer into the placenta and oxygenate the baby's blood. Left shift will cause a higher affinity for oxygen. That's when you have a high pH or an alkaline blood, low CO2, and your temperature is low. It's just the opposite of your right shift. Finally, we'll have a Haldane effect. If you bind a lot of oxygen to hemoglobin, you decrease the affinity for CO2. I think, yeah, that's CO2, that's not O2. So let's say you have a lot of O2 up here. You don't want to go down this curve to go back to CO2. You have a higher affinity for oxygen at that point. And I believe that is everything from the pneumothorax to surfactant to oxygen hemoglobin dissociation curve, as well as how blood transports in how gases transport in the blood. And I think that wraps up our discussion on that. So thank you.